solve essentially the whole concept of how DNA base pairs with itself. Unfortunately, Chargaff didn't recognize base pairing, which we'll go into a little bit. But he's a pivotal figure in the discovery of DNA and the discovery of how we think about molecular biology. His rules are still in effect today, and they're quite profound. So, if we So, as I said, today we're going to go through Chargaff. Be quiet. We're going to go through Chargaff, and at the end, I'm going to, if there's still time remaining, I want to go back over PCR again and some things about PCR I think you should have learned in lab, but based on my office hour visit, I have a strange feeling you guys did not learn certain aspects of PCR that you needed to understand. So, as I said, Chargaff was really a key figure, but in fact, DNA discovery was rather old. It's actually quite, people have been searching for what it is that is the code of life. People knew that breeding produced new life. Something must be in an egg, in a sperm of sorts to create life. I think many of you probably learned in Bio 100 the old concept that was a homunculus in a sperm. I don't, because it's a male-centric world. People know what a homunculus is. It's sort of a representation of man. There's a little tiny man in a sperm, and then as the egg developed, the man just got big. You know, it's this sort of a Baroque idea, but they sort of understood the idea. You fertilized and you created an organism, and they knew something about in that small sperm, an egg, or sperm, should I say, carried life, and they wanted to know what it was. And in the 1800s, Friedrich Miescher, in 1844, he discovered, so people had imagined that it was the nucleus of the cell that carried the information. And so he sought to figure out what it was in the nucleus of the cell that purported to have this information. And what he found was that, in fact, he found a substance called, he called it nuclein, and it was made up of protein and this other thing, this acid molecule called DNA. And remember, you have to think back to the 1800s. When you get, you know, we call it the deoxyribonucleic acid. In those days, they treated it as a chemical compound. They didn't think of it as DNA as we think of it. They think of it just like part of a chemistry set. There's this protein, and you have this acid within the nucleus, and they just remarked on it. And nowadays, it's called DNA, this acid, and of course, this particular acid carries information as we know it. Shortly thereafter, sort of around the turn of the 20th century, this scientist, Phoebus Levine, and this will show you how you can set, so there's a phrase that I always quote to my fellow scientists, and as many people as I can quote quoted to that a very famous scientist in Germany, Max Planck, said, for new ideas to come forward, old scientists must die. And that turns out to be an awful truth. And because old scientists tend to keep their ideas in publication and not let new scientists publish new ideas that might contradict those old ideas. And so this is a significant problem, and I hate to inform you guys that will go on for the rest of your lives, the same idea that once people have power, they're very reluctant to give it up. Yeah? Max Planck. So there's Max Planck Research Institute all in Germany. He's a very, very famous scientist. I think he won the Nobel Prize too. Yes. That's vaguely familiar. Physicist. So Felix Levine turns out to be a very important figure in biology because he decided to continue the work from Miescher and figure out what it was in this nucleon, in the nucleus, and in fact the nucleon. And what he did was in 1931, you see how time flies, a mere 90 years later, or a mere 30 or 40 years later, he suggested that DNA was composed of 4 components. Of course, we know those 4 components nowadays. Those are AGCT, but he had this important observation, as we'll see how important it is. Again, as I said to you, this is how science gets corrupted and how this is one of the reasons why, believe it or not, I'm trying to train you to think about biology, not as it is today per se, but to be able to think about it critically so you can see when science changes 3, 4, 5 years from now, you're able to adapt to those changes rather readily and not be forced into one particular concept. So he suggested that there were 4 components of DNA that occur in approximately equal ratios. And he said maybe DNA is composed of a repeating tetramer, which you can imagine, right? A repeating tetramer is very, very simple, right? A repeating tetramer is simply AGCT, or some version of those 4 nucleotides. And of course, if you're repeating AGCT, for example, all in a row, that is unlikely to have, right, it's hard to write a code with 4 letters. Essentially, you have one code that repeats itself. It's 1, 1, 1, 1, versus 1 and 0. It's not a lot of information here to codify anything. Even worse, Levine actually didn't even think of it as in a linear array. He actually proposed that maybe they're just tetramers on their own. So not only did Levine identify the components, he also showed that components were linked together, and so he's actually the father of the phosphate bond. So he did do something. So it's not like he didn't do anything. And he thought DNA might consist of solely a tetramer. And scientists thought perhaps proteins, 20 amino acids, they knew them at the time, had more information to codify life. Again, if you have 20, right, 1s and 0s is a lot of information, but 20 numbers is a ton of information in order to codify life. And they thought maybe this was more important. And in fact, he had this view of what DNA looked like, which was essentially a set of 4 nucleotides, in this particular case TACG, that were linked, and they're just throughout the nucleus, just these 4 nucleotides. We could think of them, for example, in nowadays terms, as like ATP. We never would think that ATP has any genetic information, so nucleotide floats around the nucleus, used for all kinds of things. You wouldn't necessarily think of that as information, but yet TACG, while a tetramer doesn't have a lot of information, and you can see this tetranucleotide hypothesis, and this is a slightly different version of that tetranucleotide hypothesis, where all 4 nucleotides are linked to each other. So again, there's not a lot of information here in order to codify anything, and he probably at the time was thinking maybe this could be an energy source for the amino acids to carry all the information. in 1945, again, now we jump ahead 20 years later or so, Schrödinger, who was a physicist, wrote a very famous book, which I think I'm going to try to buy. I'd say it every year, and I think I haven't bought it yet. Called What is Life? And What is Life actually inspired the dawn of biology research as we know it. So all the physicists, for example, like Watson and Crick, moved into biology research because of this book. They read this book, you know, the nucleus was being studied, and remember, Avery McLeod McCarty was shortly after 1945 also inspired by this. They wanted to go back and figure out what it was in the nucleus that provided life, and as we learned in Avery McLeod McCarty, it was DNA that was part of the transforming principle, and essentially DNA codified life, which means that something was amiss in the tetranucleotide hypothesis. So Chargaff noted that the publication of Avery McLeod McCarty, he realized that DNA was the key to life, and he set out to prove Levine wrong. And you know, that's a great motivation, and Rio actually was in my lab, and one of his projects was to prove some other scientists wrong. It's a terrible way of doing experiments, but it does get your motor running to do such a thing. And what he did was he realized that people were starting to separate proteins by chromatography. So they would hydrolyze proteins, run them on columns, run them on paper chromatography, and they could see the amino acids separate on the chromatography paper. And they could say, okay, this one is alanine, this is glycine, blah, blah, blah. And he decided to co-opt this technique and use it to separate out all the nucleic acids in the nucleus. And this is part of the paper that I have you read. And so what he was able to do is, from this chromatography, he was able to separate adenine from guanine, hypoxanthine and xanthine could be hydrolysis byproducts, could be. They may not be, they might be real. Uracil, cytosine, thymine. So they were able to do these under different types of conditions. These are different reagents that they did it, and they could separate those nucleotides and say, okay, I now can measure the total number of A here, for example, and I can measure the total number of T here, for example. I can measure the total number of C with this technique, and they could actually make very, very accurate measurements of how much AGCT is in the nucleus. Remember, the Levine hypothesis was that all four nucleotides were present at the same quantity. And that led him to this idea of the tetranucleotide hypothesis. Chargaff wanted to know what is the exact quantity of each of the nucleotides in the cell. And what he found was the following. If he prepared from ox, for example, thymus and spleen, and liver, and he started figuring out what was what, he noticed something rather interesting. He noticed that the percentage of adenine, for example, always equaled the percentage of thymine. And one of the things you should notice, you'll notice this early on, which somehow miraculously cleans itself up. Guanine, for example, here, slightly different from cytosine, here and here, here and here, and spleen as well, but then suddenly gets very cleaned up by later experiments. Here, this is man. Now adenine and thymine are equal ratios, and guanine and cytosine are as well, from different types of DNA, sperm, thymus, liver, carcinoma. And here, this is from microbial DNA, again, adenine is remarkably similar in quantity to thymine, and again, guanine and cytosine. Does anyone know offhand? I couldn't find this in any reading, but I was told by Rivka Rudner why this discrepancy. Does anyone know offhand why there was this discrepancy early on and it disappeared later? I'm allowing guesses.
Could it have been something having to do with the technique used?
Let's say more biological. A biological reason why he could have missed.
Could it just be point mutations in the DNA, or?
Point mutations, again, he's looking at all the nucleotides in the cell together. He wouldn't identify any point mutations, yeah? No, it's not GC binding, that's a good idea.
Methylate cytosine wasn't detected?
Methylene cytosine was either not detected or not added in to this cytosine value. Whereas, so Rivka told me that the early experiments Shargaf did, he negated to add the methyl cytosine values, but then added them afterwards. And one of the things you should note is that means you have a fair amount of methyl cytosine in your DNA, quite a bit. And as everyone knows in this room, that this is essentially the field of epigenetics, is how DNA's methylation gets modified over time, over generations, etc. So this is really a sizable percentage of nucleotides in your genome, but again, as I said, it got cleaned up and you never actually see these ever reported, and what you actually see is the following, I think it's the following. Yes, you see the following, and these are part of Shargaf's rules that I remember I had to memorize. That doesn't mean you have to memorize, but I had to memorize them in school. There were 3 rules, and I thought they were silly rules to memorize, but I think the observation is important. So again, what he noticed is that purines equal pyrimidines in value, which makes sense, right? Because if the A value equals the T value, then the G value must equal the C, and this is just basic mathematics. A plus G equals C plus T. So purines equal pyrimidines in concentration in the cell. And this is just mathematically shown in another way, is that the purines divided by the pyrimidines has a ratio of 1. Apparently this had been found by, again, so it just shows you no one ever talks about Mirsky's rules. We always learn about Shargaf's rules, and this is a common phenomenon. If you realize calculus was, for example, invented by someone else at the same time as Newton, but Newton gets all the credit for calculus. Yep.
In the liver cells, it's only showing purines.
Oh, because not every time they did a purification did they get good results. So they only reported the results that they could get. Remember, it's unpublishable in nowadays standards. Yeah, one of the things that makes it unpublishable here, I don't know if you can see, today's standards of recovery here, this would be unacceptable to have a 12% loss in recovery. No one would ever publish this. They'd say, oh no, the data's not good enough, you can't publish it. But again, this is the 40s, and those days you didn't have access to all the techniques that we have today. So the question is that, so again, Mirsky found it, but no one seemed to care. Even he found it a little bit earlier, no one cared, because unless your eyes are open, and then this is actually one of the very remarkable things about science, the literature is full of information. Full. People are publishing at an enormous rate. Do you really think I read the literature that well? I read it very, very poorly. No one can keep on top of the literature. There's just too many papers being published. And so if somebody really decides to read the literature by themselves without doing a single experiment, you can come up with a hypothesis on how something works, and it could very well be right, because there's that much data out there that people are not paying attention to. So, again, this is just one of those examples. If it's out there, no one's thinking about it at the time that this has important value, so people ignore it. But he made a big deal of it. He was probably at a better university, and so these are called Shargaf rules, not Mirsky rules. So more curiously, he really cared a lot about that the AG and TC ratios are always similar to each other at the time. So one of the things, right, of course, you all as scientists or biologists in the 21st century, you go, of course, it's AT binding and GC binding. How could he miss it? I mean, this is the most ridiculous thing. And so in fact, you can see many quotes of him throughout time. This is before DNA was figured out by Watson and Crick. He goes, the significance of these relationships is puzzling. A comparison of molar proportions of the bases reveals certain striking but perhaps meaningless regularity. he continued to write, noteworthy, although possible, no more than accidental. That all these ratios, purines and pyrimidines, were not far from one. More should not be read into these figures. These are direct quotes from him. This is why he did not win the Nobel Prize, by the way. It is noteworthy, and again in 1950, noteworthy, it's nothing more than accidental, cannot be said. All nucleic acids examined thus far at molar ratios, copurine purines, also adenine, as the number of examples of such irregular increases, the question has become pertinent whether it is merely accidental, whether it is expressed of certain structural principles that are shared by many DNAs, despite far-reaching differences in the individual comparison, the absence of recognizable periodicity in their nucleotide sequence. Then he added, it is believed that at the time has not yet come to attempt to answer. And do you know why he was thinking like this? I mean, I think it's on the next slide. He didn't want to be wrong like Levine. Because again, Levine's ideas stayed around for 20 years. 20 years of a wrong idea. He set out to disprove them. He immediately did because he saw the AGTC ratios were not the same. They were not 25, 25, 25, 25. He found this beautiful relationship between A's and T's and G's and C's, but he was so reluctant to be wrong, he actually made the extra effort to say, don't read into it, it's just not Levine's, it's just Levine is wrong. And this is actually one of the real reasons not to do experiments or not to do science, just to prove somebody wrong, because you may miss the big picture. You try to prove something rather than disprove. You have an idea and you go out and prove it, rather than you have an idea that you just want to kill. So, two reasons why he would have been wrong. What if there was some other reason the AT-GC ratio was the same? And one thing I told you, for example, here, so the first reason he might have been wrong is here I told you guanine and cytosine were not quite the same. First reason, and we had a person in the crowd who said that's because there's also methyl cytosine. So in fact, he could have been thinking, okay, there are 5 nucleotides. I don't know what to make of the methyl cytosine. So if I'm right that G pairs with C, that means G is sometimes pairing with methyl cytosine, and I can't explain it, so therefore I'm not going to suggest that that is why it's like that. So that's possible one. Possible reason two, what if there was some reason that the AT and GC ratios were the same, which we're going to talk about in the next slide. And three, remember in the very first slide I told you about hypoxanthine and xanthine nucleotides. What if actually those nucleotides have value too? Are all nucleotides in your genome AGTC? No. Certainly during replication you get U's incorporated that have, because you have RNA put in. There are lots of things going on. They found 2 other versions of methyl A in your genome. There's actually another form, I think, of methyl C in the genome. There's a lot of different versions of the nucleotides going on. And remember, in 1940s, if you didn't know anything, you might have been very cautious. However, it's unbelievable he didn't even suggest it. I mean, this is the real killer. There's no penalty in suggesting that base pairing could be the spice of life. There's no reason you not say that. You don't have to say I proved it. Just say that this suggests A binds with T and G binds to C. That's it. You don't have to be anything. So I encourage you in life, if you have an idea that seems to be right, don't afraid being called wrong. Just say, why can't it be like this? Mm-hmm.
Did he ever look at the ratio of A to U?
He didn't. I think there's U in there, but I don't think he looked at it. U is a very small percentage. Remember, might be only found in residual RNA that's been from replicating DNA.
How did he isolate the free nucleotides to make sure that they weren't
Because he hydrolyzed them. That was the very first figure. He hydrolyzed them with different techniques, and then he ran them on paper chromatography, and you can see the individual nucleotides chemically. Yep.
In the other slide with the thymus, in the second and third experiments in the thymus, the T doesn't look equal at all to the...
Right. So, well, there's the
No, the other one in the first, in table 2 on the first side, on the left side.
28,
No, on the left side, yeah, there.
24 to 28. That's right.
And
That's
the
right.
30 again.
Yeah. I mean, so there's variation too. So could certainly have been cautious, but you know, when you did the, you know, if you were to do the, if you were to average these experiments, it would look remarkably similar. I mean, one experiment doesn't prove a hypothesis. It takes a nice averaging in order to get it right. And these days, you have to do something 5, 6, 7, 8, 9 times in order to be able to get an average that's publishable. They won't let you do an experiment 2 times, 3 times to publish it. So again, so here, ATGC ratios. What if there was some other reason that this occurred? And I'll open up to anyone. What could be another reason that this occurs? Yep.
Have an
ATGC
ATGC dimer.
dimer, that's possible, I guess. Some bizarre conformation. Yep.
Maybe A and T are like matter and antimatter, and if there's an imbalance, they destroy each other.
She's in the right direction, but it has nothing to do with matter and antimatter.
Well, I guess you could think like maybe the T came from A and the C comes from
So
G.
she's getting closer. What if it's metabolism, right? What if metabolism rigs it so that the nuclei are just unhappy if you have an imbalance of G and C or A and T? That if something has nothing to do with base pairing, has to do with how you produce A's and T's and G's and C's, and this is the homeostatic balance of the nucleus. Yep.
If it's required for any biological processes, you might need A and T at similar rates and G.
That's right. That's right. So again, metabolic, something metabolic. And so, but again, of course, it's now in retrospect, right? Think back. We knew DNA codified life from Avery McLeod McCarty. 1 year later, you're presented with this knowledge that A and T are the same ratios, G's and C's are the same ratios. For you not to even speculate, and you'll see why this has become more critical, because 1 year later from there, DNA is the structure of DNA is solved very shortly thereafterwards, and it's solved by a geneticist. And you'll see why Chargaff missed it, because he wasn't thinking genetically.
Did they use his data?
Sure, absolutely. This is the irony is this is the founding one of the founding. I'm giving you the founding principles of molecular biology. The Avery McLeod McCarty paper, the Chargaff paper. The next week we'll do DNA crystallization paper, or maybe we'll do it Thursday if I try to do it Thursday. So ATGC, what if they were the same and we talked about metabolism? And one of the curious findings that I present every year, and again, you know, the reason I teach it this way is because when I decided to teach Chargaff, I started reading up on it. And I was actually really shocked that he missed it. I didn't understand it. So I started reading up on why did I just looked up papers, why did Chargaff miss it? And I put a couple of them online for you. And one curious result rather shocked me. Again, let's just take a moment. Right? If you were to write A, T, G, C, G, G, A, T, 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 A, C, G. But again, the A and T ratios would be equal because of this to this, right? That's base pairing, correct? the GC ratios are the same here, correct? Notice in this particular very short stretch, the top strand has 1, 2, 3 A's. The top strand has 3 T's. The top strand has 3 G's. And the top strand has 1 C. The bottom strand here has 3 A's, 3 T's, 3 C's, and 1 G. So again, in this very, very, very short stretch, A happens to equal T, but G does not equal C. Right? A equals T, but G does not equal C on the top strand or the bottom strand. Again, we're only looking at 10 nucleotides here. Correct? So, when DNA sequencing started coming about, a rather shocking result was seen. That if you were to look at the top strand for the 16 chromosomes, so top and bottom strand give you the same information. I don't know if you could see that G and C would be off here and G and C is off here. Doesn't matter whether you're looking at the top or bottom strand, you would see that there's an asymmetry. However, when they looked at by DNA sequencing and they did an accounting of nucleotides on the top strand versus bottom strand, which wouldn't follow ATGC base pairing rules, they found a remarkable similarity. That if you were to look at GCs, for example, in this chromosome, it's off by, what, 120 nucleotides? And here it's off by, what, also another 100 nucleotides? So the A number and the T number is remarkably similar, even on the top strand. That would not be predicted. Right? I just wrote out some arbitrary sequence here, short stretch, but it turns out the longer you write that stretch of nucleotides, it turns out there's something in the genomes for very, very long stretches. You notice these are thousands. Here for 70, 140 plus 90, 230,000 nucleotides of top strand sequence, you have parity. You have C's and G almost identical, and you have A's and T's almost identical. Is this true for all the chromosomes? It's actually almost true for all the chromosomes. It's actually the most remarkable thing. Here you're in the 150,000s for C's and G's, and here you're on 250,000s for A's and T's. Here, I'll just pick a random one, 127,000, 125,000 for G's and C's, and here in the 200,000s. So this looks remarkably like what Chargaff was afraid of, that there was something just metabolically that rigged you to have those number of nucleotides that happen to be, this represents some steady state that the cell likes to keep it at. He would have been wrong. He would, because here, no one would predict. If I showed you these numbers from the top strand, you would say, okay, then it's not AT binding. There's no way you would have said that, because it's all top strand sequence. Yeah.
But when you were putting the nucleotides, you were actually coding for amino acids, or it was just random?
Those nucleotides are just for random nucleotides. These are just random nucleotides I just wrote, right? Here I just wrote random nucleotides, ATG. This top strand, bottom strand, and if I inventory the top strand, 3 A's, 3 T's, A equals T, 3 G's, 1 C. G does not equal C. But it turns out if you do this accounting over huge stretches of genomic DNA, for some reason,
It seems very unnecessary.
right, why would that be like that? But I think this is the kind of thing Chargaff feared. But you shouldn't let fear get the best of you. You should just go ahead and publish what you think, or say what you think when you have an idea. But again, you can see that, and this actually says about how complex biology is. You would never guess this. So bottom line is, when I see this kind of data, this says to me, the reasons that keep AT binding, base pairing at the same percentage also favors it on the same top strand, because somehow metabolically it must be favored to have everything at the same AT ratio and so everything at the same GC ratio. Yep.
Is this fairly true across different life forms?
I think so. I can't swear to it. I mean, they've done it for yeast. I can't imagine it's not true for other organisms. Remember, you have to look at a big enough stretch to make this accounting valuable. You see, for 10 nucleotides, it would fail. For 100 nucleotides, it would fail. You can only see the byproduct metabolism for thousands and thousands of long stretches of nucleotides. Yep.
Would a series of C's on the top, like a really huge series of C's on the top down and unbalanced chunk, would that cause the DNA to shape differently? So I'm picturing like if you scoot off to one side and scoot back, and sort of have to keep balance, the whole thing doesn't stop functioning.
But there are long stretches of C's in the genome. Remember, this is just a pure accounting. There's no one, I mean, there could be a stretch with 100 A's in a row in it. But it means that for every stretch of 100 days in a row, there are plenty of T's to compensate on the same strand. It's a remarkably striking result and just shows you how easy it is to be fooled, where in a way you not suspect. And one of the reasons I like to present this, because you should think very clearly about biology. If this is true, it says that the nucleus in the cell is hardwired in some way to keep that AT ratio correct, that you're not making more A's than T's. It's rigged that way, and probably the fact you're making the same amount of A's and T's in the nucleus probably keeps this like this. So this is probably a reflection of some fundamental underlying biology going on in the nucleus in order to maintain the integrity of the DNA. And I think, so here this is just another slide showing you now, of course, a very nice polished version of Chargaff data, where you can see that bacteriophages, which are double-stranded, follow the same rules. Bacteriophages here follow the same rules. E. coli, and I don't have it here, but all of these double-stranded organisms follow all the perfect rules of Chargaff. All have roughly the same percentage of A's and T's and B's and C's. I should note that he's lucky he didn't do a single-stranded DNA virus or anything that happened to be single-stranded. Of course, he didn't know anything about double or single-stranded at the time. He just is, and again, that would also be cautious because we just saw, we look at a top strand, which is technically single-stranded, we also saw the same ratio. So that would be another way to know that, oops, it's not double-stranded. Yep?
Do they see the same ratios in the single-stranded?
No. So when you do single-stranded, but again, most of the single-stranded organisms are small. So again, out of 10,000 or 1,000 nucleotides, you're going to see disparate ratios. You have to go out quite a bit. And I don't know what the largest single-stranded organism is, but maybe if it's long enough, it actually has the same ratios. I don't know. So I think that's the last slide. If you guys can go, or I can go through a little bit about PCR. Okay. So one of the things I want us to talk to you guys about PCR is that I think you're missing a very subtle point. And we can continue on Thursday with this as well. But one of the points that I think you're subtly missing is that we put in DNA. You did PCR reactions in class. You put in your genomic DNA. It is double-stranded, of course, and you put in also oligos into the mix. These are short sequences, either top strand oligos or bottom strand oligos that you put in. And again, as I told you, when these DNA strands separate, this will bind down here. If you use this oligo, it would bind over here. This oligo would bind up here. This would bind up here. That's by hydrogen bonding. This was your first exercise, the homework that I gave you in class, for class. And we'll go over again hydrogen bonding on Thursday when I go through DNA. But one of the things I think you guys don't fully appreciate is the cycling of PCR. The first step, 95 degrees C, is to separate the strands. So at 95 degrees, the hydrogen bonding of double-strand DNA cannot be maintained, and no matter how long the DNA structure is, and the DNA will separate. At a certain temperature, it will reanneal. So, for example, as we shift down to 55 degrees C, for example, these DNA strands will reanneal by hydrogen bonding. Yeah?
So we've already prepared it where all of this has been
No, the DNA, so let's say, for example, it's coated with histones or whatever. At 95 degrees, all the protein loses its folding properties and becomes linear, or even starts breaking. So all the proteins are gone from the DNA at 95 degrees. Essentially, naked DNA, it's naked single-strand DNA. At 55 degrees, these DNA, genomic DNA strands, will reanneal. However, you also have oligonucleotides in them. And in fact, ironically enough, since this is very long and the oligos are very short, remember these are roughly 8 to 20 nucleotides, and these genomic regions are quite big, the genomic DNA will reanneal at a faster, better rate than the oligos will. They have much more to base pair with. What stops that from happening is that we put a ton of oligonucleotides in relative to the amount of genomic DNA that's present. We call that a molar excess. In fact, we don't just call it a molar excess, we call it a vast molar excess. It is such large quantity of oligo that we outcompete, this is biochemistry, we outcompete for the binding. And while maybe the DNA will go back and bind to itself everywhere but where the oligo is, the oligo will bind. And so it either binds like this, and the rest of the genomic DNA binds itself, but the oligo will always outcompete for binding over the genomic DNA coming back to itself. It binds based on several factors. So here I wrote 55 degrees. It binds based on several factors. The oligo binds based on length of oligo, GC content of oligo, temperature of annealing, salt concentration present. So all of these things, which are the salt concentration is your buffer, temperature of annealing is in the PCR machine, GC content and length are under scientist control. So all these things dictate what temperature you try to anneal your oligo at. So an 18 mer and a particular GC content will anneal somewhere around 55 degrees C. So most 18 mers do, and there are plenty of programs online that can do that calculation for you, depending on how many GCs are there. If you have a 20 mer, again, if you have a middling amount of GC, it's going to melt at a higher degree. That means that you don't have to go down to 55. You can just go to 60 degrees, and you will get reannealing of your oligo. Yeah?
Why is the reannealing temperature called melting temperature? Like, what's melting?
So the DNA, so melting temperature is a chemistry term where I think it defines the 50% likelihood of having an event occur where it comes together versus not occur. That's, I think that's a chemical definition, chemistry definition. Yeah? Right. Well, I know I put that in my notes, but she was, I posted it as well. So for example, and just one last point, if you have a 25 mer, it would be even higher, let's say 72 degrees C. So that means that we don't have to always go down to 55. And again, the only reason that the genomic DNA doesn't reanneal is because we have such a huge molar excess of oligo. And I want you to think about one question for Thursday, not on the quiz. Why do we use Taq polymerase versus a regular polymerase? Well, it's just I want everyone to think about this at home, right? But it's not on the quiz. We'll continue this discussion.